this lecture, we'll resume the discussion about thyroid hormone biosynthesis and transport. Just a brief summary about what we have mentioned before. This is tyrosine, which is the building block of thyroid hormones. Iodination at the 3-carbon position will produce 3-monoiodotyrosine or MET. Further iodination at carbon atom number 5 will produce 3,5-diiodotyrosine, DIT. This diagram depicts thyroglobulin molecule with 2 iodotyrosyl residues that have been brought into proximity by the tertiary and the quaternary structure of thyroglobulin. A few steps then take place to have the 2 molecules linked as a quinol ether intermediate, followed by splitting of the quinol ether, and iodothyronine and dehydroalanine are formed. So now we ended up with iodothyronine. This is called coupling of iodotyrosines to form iodothyronines. This is an oxidation reaction, and it is catalyzed by thyroperoxidase, and it takes place on the apical membrane of the thyrocytes. The major iodothyronine that is produced by the thyroid gland is this form, tetraiodothyronine, 3,5 tetraiodothyronine. The definition of a thyronine is 2 tyrosyl moieties linked together by an ether linkage. This oxygen represents an ether linkage. An ether linkage is defined as an oxygen group linking 2 alkyl or 2 aryl groups. A small amount of 3,5,3' triiodothyronine or T3 is also produced by the thyroid gland, and this is the most potent form of iodothyronine. It's mostly produced in the periphery by deiodination of tetraiodothyronine or T4. Here we depict the chemistry and interconversion of the thyroid hormones in different tissues. The most abundant form of thyroid hormones produced by the thyroid gland is T4 or thyroxine, which is 3,5 tetraiodothyronine. So this is the most abundant form. In peripheral active target tissues, iodination at 5' position is lost. This reaction is catalyzed by a specific enzyme, and this produces T3, which is the most potent form. On the other hand, T4 or tetraiodothyronine is converted in inactive tissues, for example in fasting adults, into an inactive form of thyroid hormones which is called RT3. Deiodination here takes place at position 5. So RT3 is 3, 3 prime, 5 prime triiodothyronine, and it is the inactive form. So it depends on the activity of the tissue. T4 can be converted either into the more potent form, which is T3, by deiodination at 5 prime position by a specific 5 prime deiodinase, or when the tissues are inactive, for example in fasting adults, T4 is going to be converted into an inactive form which is called RT3 by deiodination at position 5 by a specific 5-deiodinase enzyme. Now we are going to talk in some details about the synthesis and secretion of thyroid hormones. This figure represents a follicular cell or a thyrocyte. Here is the apical membrane. So the extracellular interface here is represented by the follicular colloid, while on the other side, this is the basal membrane here, the extracellular fluid is represented by the blood. The first thing which we should start with is the receptor. This receptor on the basal membrane is TSH receptor. TSH is a hormone produced by the anterior pituitary, and it binds to its receptor on the basal membrane of the follicular cell. Thus, it performs two functions. First, it promotes the growth of the thyrocyte. Second thing, it promotes the biosynthesis of thyroid hormones. Every single step in the biosynthesis of thyroid hormones is stimulated by the binding of TSH with its receptor. We're going to talk about that in some detail later. The second point we are going to talk about is the active transport of iodide across the basal membrane. So iodide moves from the extracellular space into the cell, so it moves in that direction. So this is the active transport of iodide across the cell membrane. The active transport of iodide across the cell membrane into the thyroid cell is called iodide trapping, because it is a one-way movement. Once iodide enters the cell, it is trapped inside. And it is an active process because it takes place against the concentration gradient. The thyroid gland can concentrate iodide to about 30 or 40 times more than its concentration in the blood. So this process must take place in an active way. And the system which is responsible for that is called sodium iodide Symporter. This Symporter carries out the movement of both iodide and sodium ions from the extracellular fluid, the blood, into the cell. For the iodide, this is a one-way track. Once iodide enters the cell, it goes towards the apical side. So sodium iodide Symporter transports iodide across the basal membrane, and it derives its energy from sodium-potassium ATPase, which drives sodium back into the extracellular fluid in exchange for potassium. So the sodium iodide Symporter, the NIS, transports iodide across the basal membrane, and it derives its energy from sodium-potassium ATPase. And it allows for a concentration of iodide about 30 to 40 times the concentration in the blood. This step, the active transport of iodide across the basal membrane into the thyroid cell, which is called iodide trapping, is stimulated by TSH. This step is suppressed by large amounts of iodide, because large amounts of iodide are going to affect the Symporter, both at the gene expression level and also as far as the enzyme activity is concerned. Excess iodide leads to inhibition of NIS, Symporter, both at the level of gene expression goes down, and the enzyme activity also goes down. There are certain drugs which block the Symporter. Perchlorate, for example. It competitively inhibits the Symporter. In other words, it competes with iodide for the Symporter. So to recap with this point, NIS transports iodide across the basal membrane, and it derives its energy from sodium-potassium ATPase, and it allows for concentration of iodide 30 to 40 times the concentration in the blood. Most important is that is stimulated by TSH, like all other steps of thyroid hormone synthesis, and it is suppressed by excessive amounts of iodide, because this simply inhibits the Symporter by reducing its gene expression and also its enzymatic activity. There are some drugs which block the iodide trapping, like perchlorate, for example, which competes with iodide for the NIS. So it is a kind of competitive inhibition of the NIS. Then iodide crosses the apical border to the surface of the apical membrane. This transport across the apical membrane is carried out by another transporter, which is called pendrin. Pendrin is an iodide chloride transporter. On the surface of the apical membrane, iodide is oxidized. So the first step on the surface of the apical membrane is the oxidation of iodide. So the fact that it is oxidized means it is going to lose electrons. This process prepares the iodide for the next step, which is iodination of thyrosyl residues of thyroglobulin. Thyroglobulin is synthesized inside the cell. The gene for thyroglobulin, like all other proteins, is transcribed, and the messenger RNA goes to the rough endoplasmic reticulum, and the protein is synthesized on the ribosomes, and it is secreted outside the membrane. So this is thyroglobulin, and of course it has tyrosine residues So these become iodinated after the iodides become oxidized, which means they lose electron. This prepares the iodide for the next step, which is the covalent bonding with thyrosyl residues of thyroglobulin. So it is a covalent bonding, means sharing electrons. In other words, the electrons which have been lost here are compensated for by this covalent bonding with tyrosine, which adds shared electrons between the thyrosyl residues and the iodide. The process of iodination is sometimes called organification. Then the third step after the iodination of the thyrosyl residues of thyroglobulin is the so-called coupling. This means linking pairs of iodotyrosine within the thyroglobulin, the process which we described before, to form iodothyronins. And the iodothyronins which are formed are, by and large, T4 or thyroxine, and to some extent T3, triiodothyronine. So just to summarize, these three processes which take place on the surface of the apical membrane after the transport of iodide across the apical membrane by the pendrin transporter, which is an iodide chloride transporter. Now iodide is on the surface of the apical membrane. Iodide undergoes oxidation, which means loss of the electrons, in order to prepare it for the second step, which is the iodination of the thyrosyl residues on thyroglobulin. And this is a covalent bonding between the thyrosyl residues and the iodine. Then coupling takes place. Coupling means linking pairs of iodotyrosines within the thyroglobulin. The three steps of iodide oxidation and thyrosyl iodination and coupling of iodotyrosines to form iodothyronin, the three steps are catalyzed by one and the same enzyme, thyroperoxidase. TPO catalyzes the three steps of oxidation, iodination, and coupling. Thyroperoxidase is an apical membrane bound glycoprotein, and it contains a heme moiety. Again, TPO is stimulated by TSH. This peroxidase is synthesized at the ribosomes on the rough endoplasmic reticulum, and from there it moves to Golgi. From there it is moved towards the apical membrane, where it becomes bound to the apical membrane. Oxidation by TPO will need H2O2. So there must be a system for generating H2O2. And this system is an enzyme that's called dual oxidase 2 or DuOX2. This is a member of the NADPH oxidase family and is located at the apical membrane of thyroid follicular cell. To recap with this point, the three processes of iodide oxidation and iodination of the thyrosyl residues of thyroglobulin and the coupling of iodotyrosines to form iodothyronins or T3 and T4 on the surface of the thyroglobulin. These three processes are catalyzed by a specific peroxidase called thyroperoxidase or TPO. TPO is produced inside the cell and it goes to Golgi and it moves towards the apical surface. This process of oxidation needs H2O2 peroxidase, and the peroxidase generating system is represented by a member of the NADPH oxidase family called DOX2, which is called dual oxidase 2. So it is a local enzyme. Calcium is a cofactor in this process, calcium ions. A few words about coupling. We are going to repeat what we have mentioned before. Coupling is also catalyzed by TPO, and it is the oxidation of two iodotyrosine residues brought into proximity by the tertiary and quaternary structure of the thyroglobulin. They become linked as a quinol ether intermediate, and then the quinol ether is separated to form an iodotyrosin, and the remainder of the molecule is called dehydroalanine. What concerns us here is the iodotyrosins, which are formed now on the surface of thyroglobulin. As we mentioned, in the process of coupling, two DIDs will form T4 or tetraiodothyronine, and one MIT and one DIT will form T3 or triiodothyronine. We mentioned some drugs which block the iodide transport across the basal membrane by blocking the Symporter, namely perchlorate. A group of drugs competitively inhibit TPO and thus block the synthesis of thyroid hormone. These are thiocarbamide drugs, and they include propylthiouracil, metimazole, and carbimazole. So there are some drugs which act as the basal membrane to competitively inhibit the activity of the sodium iodide Symporter and to reduce its gene expression, like perchlorate, for example, and in turn the entry of iodide into the cell is blocked, and in turn the synthesis of thyroid hormone is compromised. On the other hand, there are some drugs which function at the apical membrane to competitively inhibit thyroperoxidase. These drugs belong to the thiocarbamide group, and it includes propylthiouracil, metimazole, and carbimazole. They competitively inhibit TPO and thus block the synthesis of thyroid hormone. At the thyrocyte apical membrane, the colloid is engulfed in vesicles by pinocytosis and is absorbed inside the cell. Then lysosomes containing proteolytic enzymes then fuse with the colloid vesicle, and this will release T4 primarily and T3 after the protease hydrolysis. This releases T3 and T4 as well as the inactive iodotyroxines, MITs and DITs, and also individual amino acids. So MIT and DIT are deiodinated by specific deiodinases. Iodide is released and it is recycled again. So there is almost no loss of iodide, while tyrosine enters the amino acid pool of metabolism. T3 and T4 are released into the circulation. Thyroglobulin, again, is hydrolyzed and it produces amino acids including tyrosine residues, which enter into the pool of amino acid metabolism. So there is almost recycling of many of the components which participated in the process, including the iodide. So, as we mentioned, MIT and DIT formed during the synthesis of thyroid hormone are deiodinated by intrathyroidal deiodinase. This deiodinase is an NADPH-dependent flavoprotein. It is found in the mitochondria and microsomes. This enzyme is specific to MITs and DITs. It doesn't deiodinate T3 or T4, which now find their way out by diffusion into the blood. And as I said, the result of deiodination of MITs and DITs is iodide, which is recycled, used again for hormone synthesis, while tyrosine is an amino acid that enters into the general pool of amino acid metabolism.